Humanity loses about 40% of all crop production to pests and pathogens every single year. This is not only an economical problem, but it's also a societal one. In an ever-increasing world population, we must find ways to mitigate these losses and to ensure food security for everybody. This is a very ambitious aim, and for that we need to have a deep understanding of pathogen, ecology and evolution. And this is something we're very passionate about here at the Sainsbury Laboratory. A pathogen's success is contingent on its capacity to spread, and as such, natural selection will favor pathogens that are capable of spreading to new plants. That's why many of these pathogens co-opt the mobility of insects to do exactly that, further dispersing to new hosts. This is what we know as vector-borne pathogens. And vector-borne pathogens must walk a very thin line. If they are too virulent for their insect host and they kill it, they will be hindering their own transmission. And that is why evolutionary theory predicts that a vector-borne pathogen should be neutral to the fitness of their host or even beneficial, allowing the pathogen to spread to new plants. Vector-borne pathogens are very interesting because they must colonize and often thrive in two very different hosts, an insect and a plant. Selective pressures on these two hosts must be very different, and so are the outcome of these interactions, mutualistic in the former and pathogenic in the latter. And despite the importance that vector-borne pathogens have in the epidemiology of many plant diseases, we know very little about the biology of colonization and virulence for microbes that can exploit two very different hosts. My lab tries to answer all these questions by asking a fundamental one, and that is, how does a microbe adapt to two hosts? To answer this question, we must find a model system with rich natural history that is also experimentally tractable. And we have found tortoise beetles and their associated fungi, defensive microbes, to ask all these different questions. My lab tries to integrate as many areas of research as possible. We do a lot of fieldwork, not only to sample our insects and our fungi, but also to carry out experiments. And once back in the laboratory, we use a diversity of different techniques, from microscopy, molecular biology, plant pathology techniques, genomics, and also analytical chemistry. The fields of plant pathology, insect symbiosis, and natural product research have been traditionally studied separately, but my lab tries to bridge all these different fields of research with the ultimate aim of understanding pathogen biology and how does a microbe adapt to two hosts.